To generate human therapeutic protein, scientists need to isolate a fragment of DNA that contains the human gene and then insert the sequence into a plasmid. So in this lab we produce the DNA fragments and then these fragments are needed to create the recombinant plasmid para-R that produce red fluorescence protein in bacteria. So we use restriction enzymes to put to plasmids that create the DNA fragments. So this technique is called a restriction digest and the legs of fragments are determined by gene electrophoresis. So scientists use plasmid DNA to create recombinant DNA.
To clone the RFP gene, you will need the DNA from two different plasmids. The first plasmid, PKR, carries the gene that makes bacteria resistant to the antibiotic kanamycin, the RFP gene, and the promoter. The plasmid para contains the gene that makes bacteria resistant to the antibiotic ampicillin and a DNA sequence that activates the promoter when the bacteria are grown in the presence of an arabinose, a 5-carbon sugar that naturally occurs in various plant and bacterial carbohydrates. This sequence is called the Arabinosa Activator. An activator is a protein that regulates transcription of a gene by binding to a sequence near the promoter, thus enabling RNA polymerase to bind to the promoter and initiate transcription of the gene. The activator protein can also block the binding of the RNA polymerase and thereby inhibit the transcription of the gene.
If arabinose is present in the bacteria, the promoter will bind RNA polymerase and transcription will occur. If arabinose is not present, the promoter won't bind RNA polymerase and transcription won't occur. The plasmid PR also contains the ORI site for initiating DNA replication. The relevant components on the plasmids are the red fluorescence protein gene, the promoter, the ampicillin resistance gene, and the Arabinosa activator. In addition, is relevant the size of the plasmid, which indicates the number of base pairs and the sequences where it can be cut by the restriction enzymes that will be used in the lab. The sites labeled BAM H1 and IND3 represent the restriction sites for these two restriction enzymes.
The coding of the red fluorescence gene to the restriction enzyme are used in cutting the plasmid into which the red fluorescence gene will be inserted and isolating the gene for the second plasmid. Using two different restriction enzymes has several advantages. It allows the inserted gene to be oriented in the correct position on for copy down and the scenes line for the LNR, and it prevents the plasmid for recognition a circle without